Hello, everyone. Bonjour à tous. My name is Howard Huynh, and I am the new Beaty Postdoctoral Research Fellow in Species Discovery here at the Canadian Museum of Nature. And today I'd like to tell you a little bit more about myself as a professional mammologist, one who specializes in the taxonomy or classification of mammals in studying mammalian diversity. And I'd like to tell you a little bit about what that process typically entails in my line of work. So I come to the Canadian Museum of Nature with an extensive background in mammalogy, having received most of my training at the Royal Ontario Museum in Toronto and the New Brunswick Museum in St. John in Canada. And I've also received training in the United States of America at the Smithsonian Institution National Museum of Natural History in Washington, D.C., and at the Field Museum of Natural History in Chicago. I am trained as a field biologist and enjoy conducting biological expeditions all over the world, from studying bats and rodents in the cloud forests of Ecuador, to studying and collecting cave bats in Southeast Asia, to conducting biological surveys on Anticosti Island, and conducting expeditions in various protected natural areas in New Brunswick, and obviously having a lot of fun doing it. And doing field work has enabled me and my collaborators to collect specimens and address interesting scientific questions that pertain to various aspects of mammalian biology, conservation, and wildlife management. For example, right now I'm currently leading a large international scientific team reviewing the invasive species biology of eastern gray squirrels in Canada and around the world. However, with respect to describing and classifying new species of mammals, almost all discoveries come not from the field, but from the collection cabinets and drawers in research labs of natural history collections housed inside museums. And within the overlooked and sometimes underappreciated confines of a museum collection, I get to study the anatomy of mammals, that is, their skulls and their skins, in greater detail. And I like to show these and other similar photos to other people when I describe my work for two main reasons. One, I like to show that the ecomorphology of mammals in terms of shape and form is extremely diverse. And two, that the term biodiversity is not some abstract nebulous concept that we scientists like to use haphazardly when discussing biology. Biodiversity is real. You can see it, you can touch it, you can study it in great detail. And an analogy that I like to use centers on the functionality of a museum collection as a scientific resource. If you think about it, every museum specimen represents a data point, an historical snapshot or record, if you will, of a specific place and time. So you put all those millions of data points together, and a museum is essentially a working time machine. That is a catalog of the biological history of our world, both of the past and of the present. And I've had a great deal of fun using these time machines to explore the natural world in great depth and make interesting discoveries about the global mammal fauna. For example, from uncovering morphological and genetic diversity in Southeast Asian horseshoe bats, to describing new species of deer mice from Central America, and to detecting range expansions of different species of deer mice in parts of Atlantic Canada, which in turn has led to new genetic testing methods using quantitative PCR for detecting Borrelia burgdorferi, which is the spirochete bacterium that causes Lyme disease. Alongside my studies in bats and rodents, much of my research also includes studying the biology of carnivores, including hybridization of closely related feline species. Recently, with some collaborators here at the Canadian Museum of Nature, I published some observations and notes examining the morphology of bobcat Canada lynx hybrids. And indeed, I'm excited to be reunited with several of those colleagues here, like Dr. Kamal Kidas, to continue studying bobcat and Canada lynx biology in Canada. Several years ago, I had a chance to study genetic mixing of polar bears with brown bears. Here at the Canadian Museum of Nature, I will be working with the largest collection of polar bears in the world to study patterns in skull development and see if we can detect any significant anatomical changes imposed by anthropogenic activity such as hunting and climate change. Of course, you can't conduct good science without working with good people. And without question, I've been most fortunate to work with some of the best research curators in the world and learn alongside some fantastic students and young researchers, many of whom have become the new leaders and experts in the field of mammalogy and museum science. So I've hoped I've helped you understand a little bit about what describing mammalian diversity is like and how it's best accomplished with well-curated and managed museum collections. Certainly the Canadian Museum of Nature has one of the best collections anywhere in the world. And it's an honor and privilege for me to work with many of the scientists here, many of whom are world authorities in their respective disciplines. With that, thank you all for your time, and I look forward to sharing some more scientific discoveries with you in the near future. Merci beaucoup. À bientôt.